Point mutation. A point mutation or substitution is a genetic mutation where a single nucleotide base is changed, inserted or deleted from a sequence of DNA or RNA. Point mutations have a variety of effects on the downstream protein product, consequences that are moderately predictable based upon the specifics of the mutation. These consequences can range from benign, for example synonymous mutations, to catastrophic, for example frameshift mutations, with regard to protein production, composition, and function. Causes. Point mutations usually take place during DNA replication. DNA replication occurs when one double-stranded DNA molecule creates two single strands of DNA, each of which is a template for the creation of the complementary strand. A single point mutation can change the whole DNA sequence. Changing one purine or pyrimidine may change the amino acid that the nucleotides code for. Point mutations may arise from spontaneous mutations that occur during DNA replication. The rate of mutation may be increased by mutagens. Mutagens can be physical, such as radiation from UV rays, X-rays or extreme heat, or chemical, molecules that misplace base pairs or disrupt the helical shape of DNA. Mutagens associated with cancers are often studied to learn about cancer and its prevention. There are multiple ways for point mutations to occur. First, ultraviolet, UV, light and higher frequency light are capable of ionizing electrons, which in turn can affect DNA. Reactive oxygen molecules with free radicals, which are a byproduct of cellular metabolism, can also be very harmful to DNA. These reactants can lead to both single-stranded DNA breaks and double-stranded DNA breaks. Third, bonds in DNA eventually degrade, which creates another problem to keep the integrity of DNA to a high standard. There can also be replication errors that lead to substitution, insertion, or deletion mutations. Categorization. Transition/transversion categorization. In 1959, Ernst Fries coined the terms transitions or transversions to categorize different types of point mutations. Transitions are replacement of a purine base with another purine or replacement of a pyrimidine with another pyrimidine. Transversions are replacement of a purine with a pyrimidine or vice versa. There is a systematic difference in mutation rates for transitions, alpha, and transversions, beta. Transition mutations are about 10 times more common than transversions. Functional categorization. Nonsense mutations include stop gain and start loss. Stop gain is a mutation that results in a premature termination codon, a stop was gained, which signals the end of translation. This interruption causes the protein to be abnormally shortened. The number of amino acids lost mediates the impact on the protein's functionality and whether it will function whatsoever. Stop loss is a mutation in the original termination codon. A stop was lost, resulting in abnormal extension of a protein's carboxyl terminus. Start gain creates an August start codon upstream of the original start site. If the new August is near the original start site, in frame within the process transcript and downstream to a ribosomal binding site, it can be used to initiate translation. The likely effect is additional amino acids added to the amino terminus of the original protein. Frameshift mutations are also possible in start gain mutations, but typically do not affect translation of the original protein. Start loss is a point mutation in a transcript's August start codon, resulting in the reduction or elimination of protein production. Missense mutations code for a different amino acid. A missense mutation changes a codon so that a different protein is created, a non-synonymous change. Conservative mutations result in an amino acid change. However, the properties of the amino acid remain the same, for example, hydrophobic, hydrophilic, etc. At times, a change to one amino acid in the protein is not detrimental to the organism as a whole. Most proteins can withstand 1 or 2 point mutations before their function changes. Non-conservative mutations result in an amino acid change that has different properties than the wild type. The protein may lose its function, which can result in a disease in the organism. For example, sickle cell disease is caused by a single point mutation, a missense mutation, in the beta hemoglobin gene that converts a GAG codon into GUG, which encodes the amino acid valine rather than glutamic acid. The protein may also exhibit a gain of function or become activated, such is the case with the mutation changing a valine to glutamic acid in the BRAF gene. This leads to an activation of the RAF protein, which causes unlimited proliferative signaling in cancer cells. These are both examples of a non-conservative, missense, mutation. Silent mutations code for the same amino acid, a synonymous substitution. A silent mutation does not affect the functioning of the protein. A single nucleotide can change, but the new codon specifies the same amino acid, resulting in an unmutated protein. This type of change is called synonymous change since the old and new codon code for the same amino acid. This is possible because 64 codons specify only 20 amino acids. Different codons can lead to differential protein expression levels, however. Single base pair insertions and deletions. Sometimes the term point mutation is used to describe insertions or deletions of a single base pair, which has more of an adverse effect on the synthesized protein due to the nucleotide still being read in triplets, but in different frames, a mutation called a frameshift mutation. General consequences. Point mutations that occur in non-coding sequences are most often without consequences, although there are exceptions. If the mutated base pair is in the promoter sequence of a gene, then the expression of the gene may change. Also, if the mutation occurs in the splicing site of an intron, then this may interfere with correct splicing of the transcribed pre-mRNA. By altering just one amino acid, the entire peptide may change, thereby changing the entire protein. The new protein is called a protein variant. If the original protein functions in cellular reproduction, then this single point mutation can change the entire process of cellular reproduction for this organism. Point germline mutations can lead to beneficial as well as harmful traits or diseases. This leads to adaptations based on the environment where the organism lives. An advantageous mutation can create an advantage for the organism and lead to the traits being passed down from generation to generation, improving and benefiting the entire population. The scientific theory of evolution is greatly dependent on point mutations in cells. The theory explains the diversity and history of living organisms on Earth. In relation to point mutations, it states that beneficial mutations allow the organism to thrive and reproduce, thereby passing its positively affected mutated genes on to the next generation. On the other hand, harmful mutations cause the organism to die or be less likely to reproduce in a phenomenon known as natural selection. There are different short-term and long-term effects that can arise from mutations. Smaller ones would be a halting of the cell cycle at numerous points. This means that a codon coding for the amino acid glycine may be changed to a stop codon, causing the proteins that should have been produced to be deformed and unable to complete their intended tasks. Because the mutations can affect the DNA and thus the chromatin, it can prohibit mitosis from occurring due to the lack of a complete chromosome. Problems can also arise during the processes of transcription and replication of DNA. These all prohibit the cell from reproduction and thus lead to the death of the cell. Long-term effects can be a permanent changing of a chromosome, which can lead to a mutation. These mutations can be either beneficial or detrimental. Cancer is an example of how they can be detrimental. Other effects of point mutations, or single nucleotide polymorphisms in DNA, depend on the location of the mutation within the gene. For example, if the mutation occurs in the region of the gene responsible for coding, the amino acid sequence of the encoded protein may be altered, causing a change in the function, protein localization, stability of the protein or protein complex. Many methods have been proposed to predict the effects of missense mutations on proteins. Machine learning algorithms train their models to distinguish known disease associated from neutral mutations, whereas other methods do not explicitly train their models, but almost all methods exploit the evolutionary conservation, assuming that changes at conserved positions tend to be more deleterious. While majority of methods provide a binary classification of effects of mutations into damaging and benign, a new level of annotation is needed to offer an explanation of why and how these mutations damage proteins. Moreover, if the mutation occurs in the region of the gene where transcriptional machinery binds to the protein, the mutation can affect the binding of the transcription factors because the short nucleotide sequences recognized by the transcription factors will be altered. Mutations in this region can affect rate of efficiency of gene transcription, which in turn can alter levels of mRNA and, thus, protein levels in general. Point mutations can have several effects on the behavior and reproduction of a protein depending on where the mutation occurs in the amino acid sequence of the protein. If the mutation occurs in the region of the gene that is responsible for coding for the protein, the amino acid may be altered. This slight change in the sequence of amino acids can cause a change in the function, activation of the protein, meaning how it binds with a given enzyme, where the protein will be located within the cell, or the amount of free energy stored within the protein. If the mutation occurs in the region of the gene where transcriptional machinery binds to the protein, the mutation can affect the way in which transcription factors bind to the protein. The mechanisms of transcription bind to a protein through recognition of short nucleotide sequences. A mutation in this region may alter these sequences and, thus, change the way the transcription factors bind to the protein. Mutations in this region can affect the efficiency of gene transcription, which controls both the levels of mRNA and overall protein levels. Specific diseases caused by point mutations. Cancer. Point mutations in multiple tumor suppressor proteins cause cancer. For instance, point mutations in adenomatous polyposis coli promote tumorigenesis. A novel assay, fast parallel proteolysis, FAST, might help swift screening of specific stability defects in individual cancer patients. Neurofibromatosis. Neurofibromatosis is caused by point mutations in the neurofibromin 1 or neurofibromin 2 gene. Sickle cell anemia. Sickle cell anemia is caused by a point mutation in the beta-globin chain of hemoglobin, causing the hydrophilic amino acid glutamic acid to be replaced with the hydrophobic amino acid valine at the 6th position. The beta-globin gene is found on the short arm of chromosome 11. The association of two wild-type alpha-globin subunits with two mutant beta-globin subunits forms hemoglobin S, HbS. Under low oxygen conditions, being at high altitude, for example, the absence of a polar amino acid at position 6 of the beta-globin chain promotes the non-covalent polymerization, aggregation, of hemoglobin, which distorts red blood cells into a sickle shape and decreases their elasticity. Hemoglobin is a protein found in red blood cells, and is responsible for the transportation of oxygen through the body. There are two subunits that make up the hemoglobin protein, beta-globins and alpha-globins. Beta-hemoglobin is created from the genetic information on the HBB, or hemoglobin, beta gene found on chromosome 11p15.5. A single point mutation in this polypeptide chain, which is 147 amino acids long, results in the disease known as sickle cell anemia. Sickle cell anemia is an autosomal recessive disorder that affects 1 in 500 African Americans, and is one of the most common blood disorders in the United States. The single replacement of the 6th amino acid in the beta-globin, glutamic acid, with valine results in deformed red blood cells. These sickle-shaped cells cannot carry nearly as much oxygen as normal red blood cells, and they get caught more easily in the capillaries, cutting off blood supply to vital organs. The single nucleotide change in the beta-globin means that even the smallest of exertions on the part of the carrier results in severe pain and even heart attack. Below is a chart depicting the first 13 amino acids in the normal and abnormal sickle cell polypeptide chain. Sequence for normal hemoglobin. Sequence for sickle cell hemoglobin. Tay-Sachs disease. The cause of Tay-Sachs disease is a genetic defect that is passed from parent to child. This genetic defect is located in the hexa gene, which is found on chromosome 15. The hexa gene makes part of an enzyme called beta-hexosaminidase A, which plays a critical role in the nervous system. This enzyme helps break down a fatty substance called GM2 ganglioside in nerve cells. Mutations in the hexa gene disrupt the activity of beta-hexosaminidase A, preventing the breakdown of the fatty substances. As a result, the fatty substances accumulate to deadly levels in the brain and spinal cord. The buildup of GM2 ganglioside causes progressive damage to the nerve cells. This is the cause of the signs and symptoms of Tay-Sachs disease. Color blindness. People who are colorblind have mutations in their genes that cause a loss of either red or green cones, and they therefore have a hard time distinguishing between colors. There are 3 kinds of cones in the human eye: red, green, and blue. Now researchers have discovered that some people with a gene mutation that causes color blindness lose an entire set of color cones with no change to the clearness of their vision overall. Repeat-induced point mutation. In molecular biology, repeat-induced point mutation or RIP is a process by which DNA accumulates GC to a T transition mutations. Genomic evidence indicates that RIP occurs or has occurred in a variety of fungi, while experimental evidence indicates that RIP is active in Neurospora crassa, Podospora anserina, Magnaeportae gricia, Leptosphaeria maculans, Gibberellizae and Nectria hematococca. In Neurospora crassa, sequences mutated by RIP are often methylated de novo. RIP occurs during the sexual stage in haploid nuclei after fertilization but prior to meiotic DNA replication. In Neurofibromatosis, repeat sequences of at least 400 base pairs in length are vulnerable to RIP. Repeats with as low as 80% nucleotide identity may also be subject to RIP. Though the exact mechanism of repeat recognition and mutagenesis are poorly understood, RIP results in repeated sequences undergoing multiple transition mutations. The RIP mutations do not seem to be limited to repeated sequences. Indeed, for example, in the phytopathogenic fungus L. maculans, RIP mutations are found in single copy regions, adjacent to the repeated elements. These regions are either non-coding regions or genes encoding small secreted proteins including a virulence genes. The degree of RIP within these single copy regions was proportional to their proximity to repetitive elements. Rep and Kistler have speculated that the presence of highly repetitive regions containing transposons may promote mutation of resident effector genes. So the presence of effector genes within such regions is suggested to promote their adaptation and diversification when exposed to strong selection pressure. As RIP mutation is traditionally observed to be restricted to repetitive regions and not single copy regions, Fudel et al. Suggested that leakage of RIP mutation might occur within a relatively short distance of an RIP-affected repeat. Indeed, this has been reported in NCRASA whereby leakage of RIP was detected in single copy sequences at least 930 BP from the boundary of neighboring duplicated sequences. To elucidate the mechanism of detection of repeated sequences leading to RIP may allow to understand how the flanking sequences may also be affected. Mechanism: RIP causes GC to a T transition mutations within repeats, however, the mechanism that detects the repeated sequences is unknown. RIT is the only known protein essential for RIP. It is a DNA methyltransferase-like protein that when mutated or knocked out results in loss of RIP. Deletion of the RIT homologue in Aspergillus nigilans, Dimeta, results in loss of fertility while deletion of the RIT homologue in Ascabolus immersans, MASC1, results in fertility defects and loss of methylation induced premedically, MIP. Consequences: RIP is believed to have evolved as a defense mechanism against transposable elements, which resemble parasites by invading and multiplying within the genome. RIP creates multiple missense and nonsense mutations in the coding sequence. This hypermutation of GC to AT in repetitive sequences eliminates functional gene products of the sequence, if there were any to begin with. In addition, many of the C-bearing nucleotides become methylated, thus decreasing transcription. Use in molecular biology. Because RIP is so efficient at detecting and mutating repeats, fungal biologists often use it as a tool for mutagenesis. A second copy of a single copy gene is first transformed into the genome. The fungus must then mate and go through its sexual cycle to activate the RIP machinery. Many different mutations within the duplicated gene are obtained from even a single fertilization event so that inactivated alleles, usually due to nonsense mutations, as well as alleles containing missense mutations can be obtained. History: the cellular reproduction process of meiosis was discovered by Oscar Hartwig in 1876. Mitosis was discovered several years later in 1882 by Walter Fleming. Hartwig studied sea urchins, and noticed that each egg contained one nucleus prior to fertilization and two nuclei after. This discovery proved that one spermatozoon could fertilize an egg, and therefore proved the process of meiosis. Hermann Fell continued Hartwig's research by testing the effects of injecting several spermatozoa into an egg, and found that the process did not work with more than one spermatozoon. Fleming began his research of cell division starting in 1868. The study of cells was an increasingly popular topic in this time period. By 1873, Schneider had already begun to describe the steps of cell division. Fleming furthered this description in 1874 and 1875 as he explained the steps in more detail. He also argued with Schneider's findings that the nucleus separated into rod-like structures by suggesting that the nucleus actually separated into threads that in turn separated. Fleming concluded that cells replicate through cell division, to be more specific mitosis. Matthew Meselson and Franklin Stahl are credited with the discovery of DNA replication. Watson and Crick acknowledged that the structure of DNA did indicate that there is some form of replicating process. However, there was not a lot of research done on this aspect of DNA until after Watson and Crick. People considered all possible methods of determining the replication process of DNA, but none were successful until Meselson and Stahl. Meselson and Stahl introduced a heavy isotope into some DNA and traced its distribution. Through this experiment, Meselson and Stahl were able to prove that DNA reproduces semi-conservatively. To prove that DNA reproduces semi-conservatively. To prove that DNA reproduces semi-conservatively.